All right, I think we're going to start just to make sure that we are on time. So, welcome again, and I'm going to welcome you to the Michael E. Orn Emerging Technology Symposium, From Cells to Solution, Unlocking the Secrets for 21st Century Bioprocesses. This session is sponsored by the Michael E. Orn Endowment Fund, and by the Society for In Vitro Biology. And if you didn't know Michael was a very active member of the Society. He was born in April 6, 1955, and unfortunately passed away on August 27, 2012. And Michael had a great personality and a very energetic personality. He was the kind of individual that always have ideas, that always have new things that he wanted to try, new things that he wanted to share. And if you knew Michael, you really knew his energy, both professionally and personally. Like I said, he was a very active member of the Society. He was the editor of what used to be Explant from the Tissue Culture Association. And actually, he wasn't just a scientist, but as you can see, he was also an artist. He was drawing those comics that you see in here for actually the Explant. And that really speaks a lot about what Michael was. He was an individual that really had a great sense of humor, beside being a great scientist. I had the opportunity to meet Michael, of course, at SIVB, but I knew of Michael before I met Michael because I was in Dr. Widdom's lab where Michael got his PhD back at the University of Illinois. Michael got his PhD from, like I mentioned, University of Illinois, and he was working in Dr. Widdom's lab on photoautotrophic culture. And from there, then he went to do a postdoc at Ciba-Geigy, where he was one of the members of the team that was responsible for the transformation of orchard grass. From there, then Michael really started to work a lot in a field that is the molecular farming field. And in that specific field, he worked with different companies from Phyto and Biotech to Prodigene. And he was not only, like I mentioned, the editor of the Explant, that it is what is now In Vitro Report for the Society of In Vitro Biology, but he was also an active member of the Society. He covered different roles, from the chair of the section to the chair of the publication committee. And actually, Michael was the person really behind the current agreement that we have with Springer, that is the publisher of our journals. And actually, I was just in the Springer meeting prior to this meeting, and I just want to remind you that both journals of the Society are open for your submission. As a member of the Society, and even if you're not a member of the Society, we're always looking for great research to publish in our journals. Like I said, what we wanted to do with this session is honoring and remembering the legacy of Michael as someone that was really always looking for the next great technology, the next great idea. And I really think that the session that you're going to have today is perfectly aligned with what Michael represented for the Society and for the scientific community. So now, without any further ado, I'm just going to invite Pon, that is chairing this session. So he's going to give us an overview of the three great speakers that we have for this session. And once again, thank you for joining us today, and enjoy the session. Thank you.
Good morning and welcome. It's going to be a great giant session. And I'm not going to go give an intro, but I think we're going to take an opportunity and take the privilege to introduce the first speaker. And after the introduction, then she'll come back and then introduce the next two speakers. But after the introduction, that's 30 minutes for the speakers, and then 5 minutes for a couple of questions. And we will stick to the time, and once that's done, that you will have the speakers, they're going to be around, and so you have additional questions that you can talk to them directly. So I take this opportunity to welcome the very first speaker, Dr. Steve Webb, and it's a privilege for me to introduce him as a keynote speaker of this session. Dr. Steve Webb is actually CEO and the president of the Global Institute of Food Security in Canada, with over 20 years of experience in research, development, business, and product development. Steve has established himself as a visionary leader, driving innovation and spearheading multidisciplinary groups. I had the pleasure of working under Steve's leadership at Dow AgroSciences, Heritage Dow AgroSciences, for nearly a decade, where he pioneered plant-made vaccines and developed groundbreaking tools for genome engineering and then other product concepts, including the microspore editing without nucleic acids. That was one of the especially for the in vitro biology group here. His expertise spans immunochemistry, crop protection, biotechnology, now food security and beyond. Under Steve's guidance, Gibbs has made remarkable progress in addressing global food security challenges. His strategic thinking and collaborative approach have united industries, academia, and governments, paving the way for revolutionary solution. Today, he's going to talk about reimagining partnership and collaborations to stimulate innovations. And I welcome Steve, and then I want you to just join hands to welcome him. So Steve, take it away.
So first of all, thank you. Thanks, Pon. As Pon mentioned, my talk today is not going to be the next technology. I'm leaving that to the two follow-on speakers that are going to talk about some really cool stuff that we all need to pay attention to. I want to talk about the process, about how we identify that next opportunity, how we take that invention and translate it into market innovation. And I'll use some examples, some historic examples, and there'll be other talks on some of that at this meeting later during the week, and then talk about something that we're trying to do a little bit differently in Canada as it relates to global food security. First, I want to thank Pon and the Society for the opportunity to be here. I have been at this meeting before, and when I was with Dow AgroSciences, and my team has had the chance to present at this meeting many times in the past. One of the things that I need to grab is the slide changer. One of the things I want to introduce is the concept that innovation is a team sport. This is not something, the aha moment of invention might be an individual. My experience is that I've never been that individual contributor to an invention, because if you look at all the patents that I'm on, it's never alone. It's always with a team. And that ability to work with people, I think, creates the spark of invention. But to carry it across the line and make a market impact, it is through teams. Invention to innovation, so a potential to an actual market impact is hard, and it's getting harder today. And we have to think about new ways to work together, and we need to bring all of the sectors together. Pon mentioned that we at the Global Institute for Food Security bring government, industry, and academia together. In fact, we were founded by a partnership between government, industry, and academia. And again, the three sectors that need to work together to be able to bring those market-impacting innovations quicker. I want to do a little bit of a advertisement for us, if that's okay. Because again, I think one of the things that we do is we partner globally. We have a vision of a world where everyone has access to safe and nutritious food. And how we achieve that ambitious goal is through our mission. We work with partners to discover, develop, and deliver innovative solutions for globally sustainable food production. And there's three really important parts to that mission statement. Partnership. Again, you'll hear that again and again, and by the time I've done my 20 slides, you'll have heard it about 40 times, because that is absolutely essential. Discover, develop, and deliver. That represents the entire innovation pipeline. Because again, having an idea, an invention is good, but insufficient to make a difference on realizing sustainable food production. And sustainable has three important legs. Number one, economic. Number one, environmental. And number one, social. How can you have three ones? But we have, and we need them, and we've got great examples of how we've been able to deliver innovations into the marketplace that check all of those boxes. It's not a choice of environment over economics. It's all three. Our purpose is to act as a connector and a catalyst, and we advance innovation through our people. And I'm joined by our engineering biology platform leader, Ben Scott, who's just over there. This is a picture of Pete Burnett. Pete was the original developer of our sequencing facilities. And we have facilities and people that help build those connections and innovations. We bridge commercialization by working through the entire innovation pipeline, from the lab bench to working with customers, products that have been in the field for years. And then we serve as a connection bridge. And what I mean by that is, at GIFTS, we have a philosophy that we can grow the pie. Best example is our relationship with the Bangladesh Agricultural Research Council, where they originally came to us to actually meet one of our scientists, Dr. Andy Sharp, who's recognized globally for his leadership in plant genomics and phenomics, and to see some of our genomic capability. When we met with them within the first month of my being at GIFTS, we saw, both of us, the Bangladesh government as well as GIFTS, that the opportunity was bigger than us by ourselves. So we brought together other members of our ecosystem, the National Research Council of Canada, the University of Saskatchewan's Agriculture College, the Global Institute for Water Security, the Food Development Center, Prairie Agriculture Machinery, to be able to address a bigger opportunity and really grow the pie. And that's culminated in a $57 million investment by the Bangladesh government into building infrastructure in Bangladesh, but also supporting research through GIFTS in Canada. Again, we see that as the opportunity from a research and development perspective, but we also connected Bangladesh to key companies in Canada to source fertilizer and wheat, barley, and lentils to help feed their growing population. So again, we play all the way through, and we see the value of bringing those connections together, not just the science, but also that commercial opportunity. We think about the ecosystem in this kind of geodesic dome perspective and how we play. The Global Institute for Food Security has moved itself to the middle, if you will. It's also affectionately known as the Valley of Death on the innovation pipeline. It's where lots of inventions go to die. Real estate there is cheap, so that was one of the other reasons why we moved there. And the other thing is, we moved there because there's a missing gap, particularly in Canada. There's an architectural challenge to the innovation system in Canada, and we helped fill that. And how we filled it, I'll talk about it in terms of our structure. We act as that bridge and connector between business and academia, and we can work at scale with industry and with academia. This move and this new strategic approach for the Institute has led to 68 partnerships across the three sectors that need to work together: academia, government, and industry. And this is not just Canadian, this is global. And some of our newest partners are not here in the room today, but in the United States, some of the universities that we work with there. The other thing that we have done to enable partnership, and again, this is something that we need to be thinking about every time we enter into a structure, is how do we reduce the friction between government, academia, and industry? All are great at what they do. Working together can be sometimes difficult. One thing that we've done is created a not-for-profit called GIFTS AgTech Enterprise to act as a business window, to be able to aggregate intellectual property, to be able to drive the path to commercialization. This concept of the not-for-profit was created or identified in the document that created GIFTS almost 10 years ago. Structurally, how do we help bridge the gap between industry and academia? We have a matrix structure. We have 3 market-facing programs: accelerated breeding, biomanufacturing, and policy and regulation. I'll talk a little bit about those in a minute. But what's really foundational to us are our technology platforms, our sequencing, our omics and precision analytics lab, our data management and analytics group. It's the largest group we have at the Global Institute for Food Security that does bioinformatics, machine learning, and artificial intelligence applications, cell biology, plant growth facilities. And I mentioned earlier our newest one that Ben is leading, our engineering biology platform. These are all state-of-the-art at scale. To put it in perspective, our sequencing capability and bioinformatics capability is equal to the Joint Genomics Institute that's out in Berkeley. And one of the reasons we are equal is we got some help from them to scale up how we do our work through partnerships. And again, the opportunity to work at scale enables us to think in terms of units of 100, 1,000, 10,000 to be able to do research to support our various partners up and down the value chain. Our market-facing programs are important as well because they create the opportunity for new visions of partnerships and collaboration. The policy and regulation space is a new one for GIFTS, but it represents our unique position in the ecosystem in that we are not industry, we are not government, and we are not academia. We say we want to be the best of all 3, but it gives us a unique perspective on the whole system. And regulation and policy are essential to enabling innovation. And that has allowed us to have a seat at the table, both in Canada, in various jurisdictions around the world, either on behalf of Canada or at the request of these other countries, the European Union and Asia-Pacific regions specifically. One of the things that I want to talk about why we need to rethink our approach to innovation is the challenges that we face in global food security. And the reality is the challenges of innovation cut across everything that we do. We're on our way to 10 billion by 2050. In fact, we'll probably get there before 2050. We are seeing limited resources in the world. Again, less land, less water, less nutrients, et cetera, and how do we feed 10 billion people with a shrinking landmass and resources? Innovation. We see things like more dynamic environmental swings. For example, in Western Canada this year, we thought we were going to be walking into an absolute devastating drought in April, and then all of a sudden, luckily, we got a lot of rain in May, and topsoil moisture is good. In fact, maybe we broke a 7-year drought if we continue to get moisture. But we're seeing swings in temperature, and one of the things I told my wife when I got here, it was 32 degrees centigrade. For all you, that's like 87. Back home, it was 12 degrees, which is not very warm, and that is the middle, in June. So again, those opportunities to see swings that have taken place, and next week it'll be 86 degrees. So again, swings from 40s to 80s are happening a lot. We've called out conventional breeding here. And this is no disrespect to corn and soybean and everything else, but crops that we tend to eat follow a more traditional breeding process, longer and a little bit slower. We see the opportunity to address, and we'll talk about the productivity gap. And then the sustainability piece, again, that comment I made earlier about we need solutions that can address both economic, environment, and social. sustainability. And again, we've got good examples in the food and agricultural space where we've been able to deliver that. One of the things that this is a call for is we cannot rely on today's technology to address the problems that we face in the future. In fact, it's not good enough to even address the problems we face today. The USDA has a total factor productivity program going on, and we are, along with others around the world, are part of that team. And what it looks at here is the green line, solid green line, is the annual increase in agriculture and food productivity that will be required to feed 10 billion people by 2050. The brown line or gold line, and I can only work in primary color, so whatever color that line is the actual productivity results that to date. And you can see in the first 15 years, we were more or less on track to hit the ability to feed a growing world. This past decade, our rate of growth, total factor productivity, has dropped off. For the globe, it's about 1.1%, and that represents all countries from 3 distinct categories: developed, middle, and developing. And you can see the Canadian growth average in that period was about 1.4%, again, below track. And in Canada, in particular, we're projected to be at a 1% increase in total factor productivity. So across all 3 categories, the rate of gain has fallen behind where we need to be able to feed 10 billion people by 2050. This is the call to action for innovation. To put it in perspective, what does that mean from an economic impact, not just the social impact? In Canada alone, the reduction in productivity represents a $30 billion missed opportunity for farmers and the agri-food sector in Canada. So again, when we see the US has about the same productivity and performance improvement as we have across all developing worlds, there's a lot of actual economic loss that has taken place in addition to the social opportunity that we need to address. This is our interest at the Global Institute for Food Security, is not to raise the alarm, but to take this as the opportunity to reimagine how we do partnerships and collaborations. We know in agriculture, there's lots of challenges. Timelines are long, breeding programs are long, new product development is long. We are happy to see that new innovations like gene editing, like Pon mentioned, particularly in Canada, there's now a nice regulatory framework to fully deploy that technology. We need to work with our global partners to ensure that we all are on the same page as how we look at gene editing. Regulatory barriers, again, are very complex in the ag and food space. In particular, we hit human health, agriculture, and environment. And we also, as trading nations, and particularly North America, we need to ensure that we have regulatory approval for new innovations that we've made for this market to be accepted in the countries that we export to. Canada is one of 5 countries that's a net food exporter, if you will, of food. We have at GIFS a grower advisory panel, and I was talking to one of the farmers who farms about 30,000 acres of land. 93% of his crop is exported to global markets. So again, that is how we need to be thinking about the challenges both on regulatory and the importance. And again, the cost and time for new crop protection products is equally intense. When we think about innovation to advance food security, right now one of the challenges that we see is that we're more or less working and operating in silos. And again, there are examples of good partnerships that have taken place, but for the most part, everybody stays in their lane. The opportunity here is to drive that interdependency to advance inventions to innovations. So the potential, which is captured in the idea, into a product or a service that makes a market impacting innovation. And again, we need to be rethinking how we measure success throughout the entire innovation ecosystem. So the the publication that Pierre asked us all to put good quality papers in the society's journals is really, really important, but insufficient to drive an innovation. We need to be also thinking about metrics that are patents, licenses, new company startups, commercialization, new jobs and services and products in the marketplace. We've talked about this Venn diagram of the three sectors that need to partner together: universities, government, and the private sector. These partnerships really can reduce risk and time to key decisions. They can really unleash the potential of an idea to an innovation. We've seen that, in particular here in the US and globally, when Operation Warp Speed happened, where we went from a gene sequence to a product in less than a year with a technology platform based on mRNA that had never been a commercial vaccine technology before. So it was, again, really fast without compromising safety or efficacy. There's opportunities in agriculture to do the same thing, and they have been done in certain aspects. The opportunity of partnerships help de-risk. We think about it in terms of the development time and the ability to engage the broader ecosystem to avoid late-stage failures that usually aren't anticipated when the idea is created. So again, the other thing I think that's really interesting is this idea of moving to more of an innovation or a partnership ecosystem, so multilateral versus straight bilateral opportunities. A couple of success factors to make these types of work, this type of partnership work, or any partnership work, is strategic alignment across the partnership. Champions in senior leadership. These are not easy to do. There's a certain amount of energy that's required to get these activated, and there needs to be support from within senior leadership of all the organizations involved. Even the design of the projects that go into a partnership like this concept of creating mutual dependence, where I can't get to the milestone without my partner helping me and vice versa. And that mutual dependence creates more teamwork, more alignment, and creates the opportunity for new ideas and inventions that can lead to new innovations, and I'll talk about that in a bit. Building trust is absolutely essential. Frequent and strong communication at the project level, but also at the management level. To put it in perspective, typically when we have a project with a partner or partners, we have a weekly call set up, and you might think, wow, not a lot happens in a week, but it's actually kind of surprising how much happens in a week, and the need to be able, with those frequent touch points, to make course corrections or interventions to support the timeline for the program. And the ability, the other thing that's super important is to recognize that each of the organizations or partners might have different or diverse success factors. For example, many times when we think about academia, the one desire is they need to publish. They absolutely need to publish. And thinking about that right at the beginning, what is your IP and publication strategy from the beginning really reduces any friction that has happened. And in the time that I was with industry, I can only think, 23 years, I can only think of one time that we did not publish a paper. And the irony is we wanted to publish the paper, but the request from the reviewers for a lot more information than we were willing to disclose held back, as supplementary information, held back the publication of the paper. And that's like 1 in 23 years. My PhD was funded by industry. I got 5 papers out of it. So it was a great opportunity, I think, to really do innovative, inventive things. So again, but that recognition up front to make sure that we recognize that industry, government, and academia have different success factors. They're not different in the concept of in competition. They can actually be very synergistic to make these things work. When we think about how do we evaluate partnerships, the efficacy of the mechanism or the impact, did we actually create an impact in the marketplace? The efficacy, the efficiency of the model. This paper here refers to cost. I think that can be a little bit of a I think that's one perspective to look at it, but from the piece that I think is often missed is the one resource we don't have enough of is time. And the ability to go much faster is actually less, if we worry about a penny, we lose the value of the pound, so to speak. And that speed to decisions is absolutely key. And then looking at the equity of the impact. And one of the things that partnerships bring is the ability to have global reach versus regional or national opportunities. When we think about bridging the innovation gap, funding and a combination of skills are required. We have to think about this in terms of 3 C's: capacity, capability, and competency. Those are the 3 that are needed to be really able to help that innovation gap. So again, that's where we see GIFs as being able to play a role, at least in our ecosystem, with the technology platforms that we offer and the people that we have. Being able to address the market, and particularly in ag and food, there's certain business vertical challenges that are very unique that are often not perceived at the very beginning of the process, at the idea and knowledge creation phase, which is consumer acceptance. How often one of the best technologies that was developed to reduce gene flow had the unfortunate brand name of Terminator. That is not thinking about what consumer acceptance could be. Yet it was the best technology to ensure containment of gene. It never saw the light of day. And again, when we think about another phrase, and I'm going to say it and Ben's going to be mad at me, is when we think about the phrase synthetic biology. Who wants anything synthetic? That's why I'm hoping engineering biology is better than synthetic. But again, we need to be thinking about consumer acceptance in the ag and food space right at the beginning. And that's not something we typically take into account. Regulatory processes, scale up, and cost of goods are really important because, again, we've talked about the impact of inflation on the ability to even feed people we have today is really, really important. It's an issue in Canada. Cost of groceries are going up, and how do we ensure safe and nutritious food for everyone? Again, those ideas need to be built in right at the very beginning of the innovation process, not just the technical aspects. And Pon mentioned one of the projects that we had a chance to do at Dow AgroSciences in partnership with Fraunhofer, and David is right there from Fraunhofer, where we made the world's first plant-made vaccine. It was a complete commercial bust, and we got trapped in the misleading effects of the technical successes we had. We were able to produce it, manufacture it, and get it registered or licensed. And we lost sight of the impact on the commercial side. So again, those are things that we need to have a holistic approach to what we look at for innovation. There's unique challenges in the vertical of ag, food, and technology. Timelines are longer. You can't do 2 years of field studies in Western Canada in 1 year, and you actually only get about 90 days to do a field study in a year because winter is long. Regulatory bottlenecks before, public acceptance issue, particularly at the late stage. There's a limited pre-competitive space in ag and food versus other business verticals like the pharmaceutical industry, and a need to focus not just on the pure technology play like I mentioned before, and again, looking at how do we look at the value in the ag tech space is very different than pharma. Within the pharma space, there's some really nice technical milestones that allow valuations to come in. Ag and food, it's kind of all front-loaded, and we need to address all of these challenges as we move along as well. A couple case studies on how partnership and collaboration have created an innovation, and I'm going to talk about something that it's alive today, but I'm going to talk about it from the standpoint of about 7 years ago when we did this work, 7 to 10 years ago with Lineo Bio. We had a partnership, a long-term partnership with the Fraunhofer Institute in Aachen, Germany, the Institute for Molecular Biology and Applied Ecology. we were working on a number of projects within the framework of our partnership around insect target site identification, strain improvement for our natural products discovery work, protein expression in plant cells, gene targeting in plant cells. And one of the things that developed through the relationship, that intimacy that develops through a relationship and mutual dependence, is the recognition from our partners at Fraunhofer and our side that we had a problem at Dow AgroSciences at the time. Our speed to decisions in our insect resistance trait discovery program was long. We had an opportunity through technology that was being developed in different parts of the organization to speed up that process by about 2 weeks. And again, that opportunity to speed things up was seen as a real value. That was based on a plant cell-based cell-free expression technology, which is now in the market sold as ALICE, almost living cell expression. And again, one of the things that partnership and collaboration, it was not a project that we were interested in when we entered into the relationship. We had very pointed projects within the partnership, but through that collaboration, through at the project level and at the management level, and the deep understanding of the capacity, capability, and competency of all the partners around the table, this new invention of this technology was developed. It was spun out of Dow AgroSciences into the startup company called Lineo, and it was based on Dow AgroSciences' strategy that we wanted to remain focused on our market space, and any technology that could integrate across different business verticals, we wanted to participate in that by enabling it, by licensing it out. So again, Lineo continues to develop and deploy the technology. Their primary focus has been in the biopharmaceutical space for the technology, and in fact, they have been just awarded a major grant to participate in the global pandemic response, and their technology is being evaluated today. And again, that's an example of an innovation that started in ag and has a role in ag, but expanded across different business verticals that can even reach. Hopefully, we don't have another need for it in the pandemic response. Something that's a little more recent and something we're actively engaged in at the Global Institute for Food Security is accelerated breeding. Again, this is technology that was originally developed. Genomic selection was originally developed and deployed in the dairy industry in the early 2000s. The large multinationals have been deploying this for over a decade in corn and soybean breeding, but crops like wheat, pulse crops like peas and lentils, other oil seeds, they don't have access to that technology because the market is a little more fragmented. The value capture is not the same as a bag of corn, a unit of corn seed, et cetera. And we need to think about new ways of bringing that technology to the marketplace. And we see partnerships and collaboration as being able to do that. And what we've developed at GIFS around our technology platforms through our sequencing capability, our bioinformatics capability, and cell biology and plant growth facilities is the ability to provide the genotyping, the genomic selection and modeling, the speed breeding and double haploid technologies, as well as phenotyping to be able to help get access to both public and private breeding organizations for crops that are, I've used the phrase underserved, but I would maybe that's the wrong phrase. It's don't have access to the 21st century tools that other species like dairy, hogs, poultry, corn, and soybean have. Again, the combination of accelerated breeding is genomic selection and speed breeding or doubled haploid and modeling. And again, those really help increase the rate of genetic gain and also offer the opportunity to deliver new innovations into the marketplace. Where we are looking at it is in the context of forming a consortium that represents the entire value chain. So from the breeders, from the grower groups, from the commodity groups, from the processors, and the breeders are public sector and private sector. And again, being able to link all of the players, we see the opportunity to bring this together to help lift the entire sector in terms of, and the project we're working on now in this consortium is around yellow peas. And why would we want to work on yellow peas? Because it represents an opportunity for about a $25 billion new market in the place. So again, whole systems thinking. We are looking for long-term sustainability in the value chain, rapid return on investment. And again, I mentioned this in beef, or pardon me, in dairy. It has not been done in beef, and peas. So with that, I would like to say thank you for the opportunity to hopefully argue the case or make the case that we need new innovations to help feed 10 billion people. We need to think about partnerships and collaborations to make that occur. And I look forward to any questions. Thanks.
Questions? So those who have questions, please come forward to the mic here. None?
Good morning, ladies and gentlemen. My sincere thanks to Pon and Michelle for kind invitation for this In Vitro Biology. And I would like to, I'm very honored to introduce today the next speaker, Professor Dr. Bruce Falk. My mentor for 15 years, a geologist profoundly captivated by the intricate interplay of earth and life. His academic journey has taken him from the depths of our planet to understanding rock, water, mineral interactions with biological complexity. Dr. Bruce's scholarly voyage began with a Bachelor of Science in Geology from Bradley University that led him to pursue master's degree in geology and paleobiology, culminating in a PhD in geology. Postdoctoral fellowships at prestigious institutions such as Free University of Amsterdam, the University of California at Berkeley, and NASA Ames Research Center expanded his horizons, enabling him to delve into the fascinating intersections of geology, biology, and the potential for life. Has experimental stations around this planet. He has worked around the entire planet. Throughout his career, his research interests have spanned a remarkable spectrum, from the biomineralization process that create intricate skeletal structures in marine organisms, studied microbial communities thriving in extreme environments like Yellowstone National Park. His work was continuously sought to uncover mysteries, life's interaction with Earth's geological processes. These efforts have resulted in over 120 peer-reviewed publications, several book chapters and books, and contribution to monographs. Dr. Bruce Falk has publications in renowned journals such as American Association for Petroleum Geologists, Astrobiology, Nature Reviews Urology, Nature Geoscience. These papers highlight the breadth of his research program, investigating everything from biosignatures on this and other planets which are critical for future manned space explorations. He is currently focusing on the calcifications in human body, such as kidney stones, cardiovascular calcification, and breast microcalcification. He is serving currently as the Ralph E. Grimm Chair at the University of Illinois and is leading a dynamic research group dedicated to advance the field of geobiology and universal biomineralization. Dr. Bruce continues to cultivate curiosity, champion exploration, and inspire the next wave of innovators poised to unlock the mysteries of our planet, the universe, and beyond. Dr. Professor Falk.
Well, good Sunday morning thank you for rolling out so early. Appreciate it very much. Shiv, thank you for that very thoughtful and generous introduction. Pon and the committee for the entire World Congress. It's a great honor and privilege to be here speaking to you this morning, and thank you so much. I stand here as a humble geologist and born-again biologist. And I have had a journey which I could have never predicted, and it only happened because of great friends and colleagues, institutions, and industry. I've had work experience directly in industry and also funding over the years to complement the federal funding as well. I have a mantra for you this morning: life makes rock. Life makes rock. You know, it sounds weird, but then hopefully by the end of this brief presentation, you'll have an idea what I mean. Well, you know, in our bodies, every single cell, every single tissue, it has a relationship with the growth of minerals in the body. And somehow along the way, you know, plants, cactus, they're actually, many cacti are identified taxonomically based on their spines and the biomineralization. Biomineralization takes place and it can be really helpful. Bones, pretty good idea. Teeth, but also biomineralization, the growth of minerals in cells and tissues, can be lethal. The calcification of human heart, the growth of kidney stones. And these processes of biomineralization take place across great expanses of time and space. As an example, fractions of a second all the way to millions and billions of years. And then we go all the way from angstroms all the way to light years. So this is how we view this in this world of astrobiology, geobiology, and biomineralization. And so everything we're dealing with right now, and again, I'm so honored to be with these other eloquent and exciting speakers this morning. When I see those things, I'm blown away. I've already learned so much, and the Congress has only started. But I also see, to me, time and space, right? Right now is a snapshot. And everything that we see that you were just presenting so beautifully in terms of the urgent need for these partnerships, I view that through the lens of time and space. And it's not something isolated. It's a result of very long processes. And I think that's an important thing to think about. So I want to talk about the fundamental role of biomineralization in the future of in vitro biology. And I warmly welcome you. You know, this is a good way to keep people in the audience is you give them a present, right? So I brought a copy of our recent book on Yellowstone for all of you. And we brought enough, I think, for almost everybody at the Congress. And some are sitting over here at this table, and then we have some that will be sitting out the tables by the registration. So please make sure you bring a copy home with you, and please go to Yellowstone as soon as possible. And you'll see our work there in the visitor center at Yellowstone. When you walk from Old Faithful to the visitor center, take a left. There are multiple rooms with displays of our work there. I also want to thank Tom Murphy. He's a professional photographer who lived in Livingston. You might have seen his video, Christmas in Yellowstone, which was nominated for an Emmy. And Tom is another big old bull who spent a lot of time by himself in the wilderness. And he's another great partner. Just like with Shiv, we've worked for years and decades together to bring these to fruition. So please make sure you bring one of the copies home with you. So I've given you a little bit of overview. Biomineralization, there's many things you can call it, but the idea is that life depends on mineralization. And it had to deal with it from the very beginning. We'll talk about that. And it's a result of both internal and external processes, this engagement of environment, evolution, and ecology through time and space. And these life-water interactions play a role in everything that we're talking about today at the World Congress on In Vitro Biology. And I don't know where this happened, but somewhere along the way, the mineralization got left off. It got looked at as some kind of inconvenient truth that we needed to get rid of. Actually, it's a record book of process. It records physiology and metabolism at a level that will hopefully be mind-blowing to you in just a moment. So it's incredibly important. Let's bring that back into the picture as we look at all applications of in vitro biology. And biomineralization, it's such an important component. And when I say mineralization, biomineralization, I mean both amorphous phases, in other words, prior to the actual structural geometric configuration of ions, all the way through crystalline. And so that's another thing that gets left off when you say the word biomineralization. So we're going to see that in just a moment. And these fundamental processes actually end up being life or death, success or failure, for all of us as we sit here in all organisms that are alive on planet Earth. And I'd like to make the pitch that life makes rock, and we need to integrate biological and biomineralization approaches for the future of in vitro biology. This is the deep sea floor. On the left-hand side, we believe that life, that's one of the models or areas we think life formed on the deep sea floor spreading centers. And Carl Woese and Abigail Selliers at Illinois were two great friends and mentors for 2 decades with me. Carl and I actually forged that diagram on the right-hand side sitting in his living room in Urbana. And it's just an idea that the vertical, the y-axis is geological time going back to birth of the Earth at about 4.6 billion. And the idea that we think that life might have started on the seafloor via water-rock interactions, biomineralization. And when we had a naked RNA world without cell walls, actually the minerals that were reacting between basalts and seawater were making the template from which all the biomolecules of life were generated eventually from. So we can't get away from it. This is so important. And Shiv and I, we call this the eternal triangle. Life, water, mineral interactions through time and space. So that's the focus I want you to have and walk away from. And our version of biomineralization has been to work our way all the way from kidney stones through subsurface oil field and gas and reservoirs of hydrocarbons, meteor impacts, killing dinosaurs, throwing rocks at Earth and taking out T-Rex 65 million years ago. We looked at life on other planets using astrobiology analyses in Yellowstone. And then we've also worked on Roman aqueducts. We have a series of papers on that where we apply these same things, climbed up into the aqueducts throughout ancient Rome and be able to reconstruct what the Romans were doing at that time. And then use coral reefs as a sensitive fingerprint of geological and climate change through time as a way to face climate change in the future. If you kind of squint a little bit, I don't know if you realize this, but Champaign-Urbana is in the center of the universe. And you can kind of see that on this map here. So these are all the places, you know, we have a great little airport, Willard Airport in Champaign-Urbana, and these are all the places that we have active research. So we certainly are very thankful for the flight connection from Urbana to Chicago and Urbana to Dallas-Fort Worth. But we've been very fortunate. In all these places, we bring the mantra of life makes rock. We bring life-water-rock interactions with us. And so in vitro characterization is so important. What does that mean? Things like size, shape, and distribution of biominerals. Where do they occur? When do they form? How do you go from the original tissues all the way into diseased tissues that are encrusted and actually entombment? Our bodies are entombing cells as we speak within the mineralization process. And we want to look at layering. That's another really big thing I want you to think about, because a lot of people say, oh, this is sounding really good. I'm excited. I want to learn more. But what's the geology part of this? And the geology part is the layering. There's a layering history that's laid down that I'll speak to you in a moment. It's just like the Grand Canyon. It just occurs at a much smaller scale. And then the other thing about this, in terms of a roadmap, is we want to be able to look at simultaneous and synchronous and sequential reactions, going from original cells into tissues and then having the biomineralization process take place. And so, again, a time-space continuum. Think about your projects with this idea. There was a before, there's an after. You know, you kind of get into cosmic philosophy when you do this, but I think it's actually a really important part of this. And one of the things we've done is invested a lot in microfluidics. I'll show you this very briefly. But there's many ways now with the amazing arsenal of technology to push these things forward. And then we want to look at human and animal and plant and microbial interactions in both natural and engineered settings to be able to answer these challenging, vexing questions that face our society. So I'm going to start off with these pictures on the back. Dr. Mayendi Sivaguru, I think most of you know him as Shiv. And Shiv is the mastermind behind, I sit next to him. And so I'm honored. He's the one who runs the spacecraft. And this is one on our new LSM 980 microscope that we have at Illinois. But these pictures you see, this is a kidney stone. This is super resolution autofluorescence. So it's 3 to 4 channel autofluorescence. So this is a kidney stone. And what do you see? You see geometries and you see layers. That's what I want you to focus on. And here's a picture of Shiv in our laboratory. And we use multiple wavelengths, and this is just autofluorescence. We've also worked really hard with immunohistochemistry and being able to do induced fluorescence as well. But remember now, the optical world allows us, with these techniques and instruments, we can do analyses down to the scale of 140 nanometers, and even lower when you do electronic zoom. So now everything's converging. X-rays, optical, electron, lasers. All these things are converging. I remember when I was a graduate student and you had to run from one place across campus to do electron microscopy to another place across campus to do some early version of X-rays to another place to do the optical. Now it's one-stop shopping. And at Illinois, we've built this all out into a facility that I'll tell you about that we also warmly welcome you to use. It's called the Roy J. Carver Biotechnology Center. The Mayo Clinic. I knocked on their door and they opened. They never even met a geologist before. So it's always fun, when you start moving these things across these disciplines. And, the Mayo Clinic is such an incredible institution of excellence. And they supported our work with making a documentary. I won't show you the whole thing, but I'm going to show you 2 minutes of it, okay?
Probably see a couple of patients a day at the emergency department with kidney stones. I have had several women tell me that this hurts worse than labor.
What we're trying to understand on many different fronts is why some people get kidney stones and why some people don't.
I always, as a geologist, wondered, what's the geology of a kidney stone? The similarities between the human kidney and hot spring environments, such as those that occur in Yellowstone, are simply remarkable. Mayo is the kind of place where it's not a harebrained idea to have a geologist knock on the door.
Where nephrologists have worked most closely is understanding the physiology or how the stones develop.
I haven't ever met a rock that hasn't crystallized and dissolved and crystallized and dissolved and crystallized and dissolved. That kidney stone is a dynamic history of both crystal growth and crystal dissolving.
If we can figure out what's leading to the rock growing within us, we hopefully can interrupt that with either antibiotics or a drug that targets a specific protein that might be leading to the accumulation of rock.
I think it'll be a game changer really for kidney stones.
It's rare to get people like geologists and medical doctors to sit down and do something serious together in terms of some research. But when that happens, magic happens. So, fantastic partners. And we've been able to do so much because of the openness and this idea that it doesn't matter what your discipline is. It matters what you do with your life and how you contribute and support what's needed in society. And that's the mantra of Life Makes Rock. So just a few shots. You see a kidney stone up in the upper left there. That's those are X-ray images in 3D and cross-section, virtual cross-sections. And you think of just a lump that's really painful that you need to get out of your body. Actually, we cut these very thin, 20 microns thick or thinner, and you can see stratigraphy. Stratigraphy is layering. And as an example, on the outside of the kidney stone, you see there's a juxtaposition of layering. The layering is truncated, and the inner layers are cut, and then outer layers move around. That's a history of process. And look at the layers. These are layers that are forming now, we think, on minute-by-minute basis. There is no other record that we can, there's no other instrument we can put into our bodies that will record these processes at these kind of levels. And each of those layers is a history of cell metabolism, physiology, in some cases microorganisms. And this is what the same kidney stone looks like in super resolution autofluorescence. It's remarkable, isn't it? Look at the information, minute by minute, hour by hour history. So now we're working our way through the layers, through the truncations, identifying moments. When I first started working on kidney stones, the general thought was they don't dissolve much. Well, Shiv and I have found that these kidney stones actually dissolve upwards of 50% by volume. 50% partial dissolution within our bodies. So one of our now targets is how does our body do the dissolving? And then how can we slow down the growth of the kidney stones? And the answer is with things like proteins. Here's what the New York Times picked up. Again, it's just beautiful, right? But you can't believe it's a kidney stone. But the information here is just like looking over the edge of the Grand Canyon. And we've carefully now mined this information to put together a sequential history. We call it the paragenetic sequence. Sorry, it's a geoscience term, but paragenetic sequence, the historical development of anything that has layers in it. And so a fellow named Nicholas Steno in about 1663, he was a medical doctor in Copenhagen, and then on the weekends he went and looked at the rocks around the coast. And he's the one who came up with the law of superposition, that rocks at the bottom are older than the rocks at the top as long as they haven't been disturbed. That was a medical doctor transitioning into geology in the 1600s. So this is old news, right? I'm just following the process, just going backwards. And the thing I want you to realize is we know the Grand Canyon, beautiful layering rocks. Geologists know a lot about layers. And you can see there on the upper right in Utah, there's fractures, broken rock, faults, where either side of the fracture you have movement. You start seeing rocks being broken up and things. But look at those pictures down below from Greece. When you have plate tectonics and motion of the earth, you actually squeeze, contort, bend, and fold. These are things that, as geologists, we use to recreate the history of a rock and deep history. But guess what? These tiny layers and their history of successions and truncations and their distributions, we can do the same thing. So this is actually a picture of a kidney stone on the left, and the right-hand side is the sequence of events, the paragenetic sequence going from original to end. And all that information on the right-hand side gives us a roadmap where in between individual steps, going from that kind of orange-colored crystal, then you have crystal growing in the urine, then they come together and make the layers. That's all yielded in this history, and then that gives us the capacity to make targets that we go after for processes and mechanisms within the kidney stone. And so this is, again, another kidney stone that we worked on, and all that information on the right, going from the tubules within the renal tissue all the way down, that graphic novel that we put together, that is a roadmap now. We're now doing systematic testing using microfluidics to understand what proteins, what molecules, what physiologies are actually causing the growth, the dissolution, and the entire package of making a kidney stone. And you see things you don't expect. Like on the left-hand side, those are called sector zones. The light gray within the diamond-shaped crystals, those are calcium oxalate dihydrate within a kidney stone. But all you have to know for now is that those patterns of growth, those sector zones, those gray areas, they tell us that these stones were 100% growing at disequilibrium in kinetics. No equilibrium process. And you know better than anybody how important it is to know that you're in a realm of kinetics instead of equilibria. And then on the right-hand side, the lower right is actually a volcanic deposit from a volcano basalt in Kazakhstan. And we're comparing that to a kidney stone from a patient at the Mayo Clinic, identical. You have tiny spherules, you have growth, you have many things happening. And then even within your kidneys, and we're all doing this, even healthy people, we're all forming kidney stones, is that we successfully dissolve them, and kidney formers don't. And you can see there's actually fractures and faults. Would you ever expect to see a fracture, faulted, and folded rock deposit within your body? Absolutely not. But that's how our bodies operate. And then Shiv and I have been able to, and our team, I should say, have been able to look at, as the minerals grow, as these rocks form, as the kidney stone progresses, it entraps microorganisms. So not only is there a record of physiology and metabolism, but we can actually extract microbial components, polypeptides, 16S ribosomal RNA gene sequences. We can, this is all, it's a zoo that's recorded within the kidney stones. And on the right-hand side, that's the river Styx, and that's the feared Charon, the gatekeeper, the ferryman, to get people from, after death, of course, from one side of existence to the other. And we've seen now that macrophages and multinucleated giant cells actually operate like Charon, where they actually encapsulate crystals and move them from one part of the kidney to another, and then dump them off. So that's Charon operating the mysterious ferryman of the river Styx. And then, again, there's a lot of information here, but I just wanted to tell you one of the roadmaps. What does a roadmap mean to us? It's things like having the Gibbs free energy of reaction on the y-axis versus what are the changes in form, shape, mineralogy, and chemistry of the mineral deposits. We call that a diagenetic phase transition. But I'm just trying to give you a flavor of, with this roadmap in hand, this is what guides our experimentation. And part of the experimentation is the microfluidics. So this is our lab in Illinois. I liked your comment about having the right names. So we call this the GeoBioCell. Now my engineering colleagues who actually know how to do these that we work with, they call it a microfluidic device. I don't know, GeoBioCell, microfluidic device. They're both fine words, but I think words matter, right? Especially nowadays, right? We have to be very careful about how we package and present them. It's kind of like life makes rock. Or how about the eternal triangle? That sounds a lot funner to remember when you're a 14-year-old kid than life, water, mineral interactions, at least to me, right? So this is actually a picture of looking into a microfluidic device, seeing, in this case, this is a sulfate-reducing bacterium we collected from an oil field, and we're simulating life that lives in rock at about 2 kilometers water depth. Those are black biofilms, and those are grains of silicon dioxide. And then I'm just going to end with the human heart. So we just had a paper come out this last week. I'm calling that a heart of stone. What do you think, huh? So it's human aortic valve calcification, but again, Shiv and I and our partners, in this case, instead of the Mayo Clinic, it's at UCLA Health, but we have our entire team. The reason I didn't show a slide of our colleagues and appreciation, it's just I tried to make it, and we had almost 100 people on the slide, and it doesn't do much good to see it, but I just want to let you know, this is a result of hundreds of people working together tirelessly for decades to get us to this point, and we are so thankful and appreciative for that work. But I want to show you our most recent one that came out this week in Scientific Reports. So here's a human aortic heart valve in the middle there. Most people have 3 tricuspid valves, and one of the great threats, 18 million people a year die of heart disease, and a very significant proportion of that are people who are dying from having calcification of the tissues in the cardiovascular system. Again, you might say, my goodness, why on earth would our bodies be growing rocks in our heart? It's because life evolved in a rock-growing environment, and every time you have fluids and physiology and metabolism, you almost by definition have to be growing minerals. So it's not a surprise, it's actually a natural manifestation, and really the question is, why hasn't someone tried to look at this before? I think that's kind of the interesting question. Again, it goes back to, I won't ask for hands, but maybe there's a few of you in the audience who have never met a geologist yourself. So we want to change that. Make sure you get a book and bring it home. Okay. All right, so there's the heart valve, and they calcify. That's a micro CT image, and the hot spots in the micro CT is the calcification. And people grow upwards of 30 to 40% of their heart becomes solid calcified rock prior to passing. So it's a very serious problem. And then the lower right is a micro CT image of one of these deposits. And what we've seen is, and again, this is where we start drawing, we could not do our work at all on the human body now if we hadn't looked at coral reefs, hot springs, Roman aqueducts, the deep subsurface, kidney stones, and now heart. And if we hadn't done all those, we could never now be moving to breast cancer. So what we're seeing is, this is a picture, again, it's a super resolution image of the nodules of calcium-rich minerals. In this case, it's an amorphous calcium phosphate that's at the verge of transitioning into hydroxyapatite, the mineral of bone. So actually our hearts and our cardiovascular system, as well as human breast tumors, actually are starting to grow human bone in these places. But it's the early phases of it. That's the amorphous phase, and that's where most of the action is. But I just wanted to say what we've gotten out of this as an example for you are two things: that our bodies upregulate osteopontin. Osteopontin is a known promoter for the growth of apatite in bones. So a lot of medical doctors use osteopontin as a way to enhance bone growth. But what osteopontin does when you have the same chemistries, calcium phosphate, in the amorphous phase, the osteopontin protein is an inhibitor. It actually slows down the rate of growth of the amorphous form. So what our bodies have done, and this is all evolution, right? We're just a snapshot of 4.2 million years of human beings having a machine called our body looking like the way it is now. Our bodies do two things. They upregulate the osteopontin, and cleverly that slows down the growth of the amorphous phases. Thank goodness. That's why we can live to 75 to 100 years. And the other thing it does is it actually stretches and grows collagen and encapsulates collagen around the mineral growth. Collagen acts as an aquiclude. Sorry for all these geology terms. Paragenetic sequence, aquiclude, plate tectonics. Guess what? It all fits, right? And so blocking water from something, we call that an aquiclude in the subsurface. Collagen blocks water from the blood, and the blood is saturated with calcium and phosphate. So we're actually doomed, right? I mean, blood is full of the cocktail that Earth has evolved to make minerals grow that are calcium rich. So our body's done two things: make a lot of osteopontin, slow it down, and then they encapsulate and protect the mineral deposits from water. And by doing that, we can live to 100 years old. Without that, we'd be much, much younger in age. Now, again, I can't speak more highly of Shiv. Not only one of the most brilliant scientists I've ever met in my life, but a phenomenal person. I hope you get a chance to talk to him while you're here. But this is one of Shiv's images. This is a 20-micron thick histology section. In geology, we call it a petrographic thin section. You call it a histology section. But this is a 20-micron thick petrographic section of a calcified human aortic heart valve. 20 microns thick, and using the LSM 980 with a spatial resolution, at this case of about 140 nanometers, you can dive in at this scale, right? 20 microns. And so we use the this lets you map and determine where's the original tissues. What was the sequence of events of the early phases of the amorphous calcium phosphate? When do you kick into the osteopontin to slow the process down? And when do you encapsulate it with the collagen? And the collagen is on the outside of this. These are pictures of the interior of these stones that grow in the heart. And the collagen are those bright colored layers that are up around the edges. And then as the stone grows, it actually incorporates the collagen that used to protect it. And then this is another version of the roadmap. Again, I'm not trying to blind you with science, but I want to give you a very specific example. Gibbs free energy reaction. We know these things are working kinetically, but as long as you know that, then you can adjust your modeling for integrated kinetic and thermodynamic modeling, and then you importantly use the diagenetic phases, the changes. Diagenesis means physical, chemical, and biological changes during the history of something. And then we put them together in a sequence, going from original tissues. The top event 1 is the original tissue of collagen with no mineral growth, then all the way down through event 5, which is the full-blown catastrophic growth. And then what happens is that the growth of the minerals cause stenosis. It caused stiffening. And again, this is where we know from the coral reef work, corals, the soft corals that go in the ocean, right? You might have seen those on pictures, or you've already been in the water yourself on scuba or snorkel. Those soft corals, guess what? They have hard spicules, hard mineral growths, just like these deposits. And what happens is when you put spicules or hard mineral deposits into something flexible and rubbery, then when it's not flexed, when it's not having force applied to it, it's very soft, right? But then when you flex it enough and bend the tissue, those tiny particles touch each other. And when they touch each other, they magically become stiff. So I don't know if any of you have ever driven a car, but have you ever thought why car tires are black? Because it's rubber, right? For goodness sake, rubber's kind of brown, beige, translucent. They put hard, tiny particles of carbon into the rubber. And so when the rubber's not inflated, it's kind of a rubbery tire and it looks black. But you put air into it, it puts stress strain onto the tissues, or in this case on the rubber bends, and then those tiny particles touch, and it becomes hard as a rock. What an incredible evolutionary tool to make things hard, but you don't want to use that tool when you're talking about your aortic heart valve. So I just wanted to reset the stage. I hope if nothing else today, you get your Yellowstone book and you have a couple additional things to think about that you can share with others. Life makes rock. And life on Earth, its success evolutionarily and in the future, is a result of the eternal triangle: life, mineral, rock interactions. And I want to reset the stage so you start thinking about mineral growth and all the incredible systems that you folks are working on in vitro biology. Look at the minerals. When did the minerals form? How are they distributed? How do they affect all aspects of the growth of the plants, the success of the harvest, all these kind of attributes? How does the mineral growth play into that? And I think it makes a big difference in the long run. And I just want to end here by saying that please, the Carver Biotechnology Center in Illinois, we have about 36 full-time employees. We support, on the average, about $63 million of research activity each year. And we have a full suite: DNA services, cytometry and microscopy to omics, bioinformatics, functional genomics, proteomics, metabolomics. And Shiv is the director of the cytometry and microscopy to omics laboratory. And the part you might be especially interested in is within the cytometry microscopy to omics, we've built out cytometry, that's both analyzers and sorters, microscopy at the level that we've shown you, and then we have the entire suite of all the omics: metabolomics, proteomics, genomics, everything put together. And we're more or less operating now as a 10x center of excellence in terms of a cutting edge application of all that. And this is all open to the public. So please, we are there, we are ready, we stand excited to engage. Right now we have about 525 projects from around the world that we're supporting, and we warmly welcome all of you to contact us. And especially important is that now we've invested more than $3 million over the last few years into building out spatial biology. And so you can first characterize in 3 dimensions the structure and composition, whether it's plant rootlets, whether it's a human heart, what have you. We have the X-ray, Raman, we have the microscopy to be able to do that. Then we'll work with you all the way through looking at intact samples on histology sections or separating out cells, and using our cell sorters, using our analyzers, or being able to then work our way through the 10x chromium and xenium and cytosist to be able to eventually then do 50 nanometer resolution of gene expression and protein composition. So it's not like this is a thing that we're building towards. The doors are open. We just finished doing this work, and it's because of Shiv's pioneering leadership in the laboratory, and we are open for business. So, and we're used to working with all people all over the world in all applications, industrial, academic, government, and all the other flavors of what those interactions could look at. So please look us up. We will respond immediately, I promise, and we hope we can work with you. So thank you so much for your time this morning, and it's been such an honor and pleasure to be here.
My name is VT. I'm from Midwestern. What an incredible talk. And as a geologist, to just go in parallel with the life sciences. So that's really fascinating, that how these crystals are being made. My question to you is that some people are highly predisposed to get kidney stones. Probably they are either dehydrated or they have urease producer infectious disease. So my question is that it has been told or mentioned in the literature that if you have apple cider vinegar or lemon juice, actually it dissolves the crystals. So one of the things I would ask you that, what do you think about predisposition? How do you think about people who want to do as a prophylaxis that they can use something to not to have a bigger kidney stones, because that's a serious problem.
Right. Thank you for that excellent question. I want to point out, I'm a geologist, not a medical doctor. So I work with the pros. So that's a great question. I will tell you that, again, 50% by volume of these stones have dissolved partially in the process of making the stone. And we know that part of that process is you have pulses in which you get like citric acids or other things. But keep in mind that that's where the geological characterization is important, because we've found now that there's something on the order of 15 different minerals that can make up kidney stones. So a kidney stone is not a kidney stone is not a kidney stone. And some of those minerals, they actually dissolve at low pH, and some of them actually grow at low pH. And some of them dissolve at high pH, and some of them grow at high pH. So knowing what you're working with, so you need to find out which series of stones you might be facing, and then you need to craft an individual medicine plan to be able to track that forward. And one of our goals is, using this arsenal of everything we've talked about, we'd like to slow down the growth of kidney stones so much that unless you're 1 million years old, you will not get kidney stones. Now, keep in mind, all of us at this moment are growing kidney stones. It's just that, fortunately, some of us have the luxury that our bodies are dissolving it as fast as it grows. But that's not going to happen forever. So.
Hi, I'm Marceline Egnen from Tuskegee. Very impressive presentation.
Thank you.
My mom suffered from Alzheimer's and she passed.
I'm sorry.
So I'm interested in what your vision is in looking at Alzheimer's patient brain.
Yeah.
See if the same happen.
Thank you so much for that wonderful, and I'm so sorry for the passing of your mother. I can tell you too, you're right on target. So another category, we're now expanding, kidney stones, heart, breast, and then another one that Shiv and I are working on is the human brain. So the pineal gland, actually we've been finding that some people grow kidney stones centimeters in size in their brain. And so the amyloid system is one of the major ones. And there's actually been some recent work where they've looked at amyloid and correlation of that with the growth of the cardiovascular calcification. So we believe that these processes are linked. And one thing I didn't point out is that you have organic minerals and you have inorganic minerals, but both are playing at the same time. And so we think that these this is just, this is like the first map that a guy named John Coulter from St. Louis drew of Yellowstone when he was out there in 1802, right? He drew a very nice map, and he got it hand by hand back to St. Louis, and it was published by Washington Irving, of the Headless Horseman. They published that map, and they had a crude sketch of what Yellowstone looked like. And it took another 100 years for it to get mapped out. What we're trying to present here with these roadmaps of reaction, kinetics, equilibrium, and changes of mineralogy and phases is that crude first map of Yellowstone. And we think that this is going to be worked out then into 3 dimensions, and the amyloids and other types of organic processes are going to be built into that simultaneously. So thank you.
That was a fascinating lecture, Dr. Falk. I'm Shirley Pomponi from Harbor Branch Oceanographic Institute. So I have a ton of questions. I'll only ask you 2 quick ones. One is, have you looked into osteoporosis, so the reverse?
Yes.
And then also, what about biosilicification, so silica versus
Thank you so much. And I want you to know, I had the honor, I taught a summer course at Harbor Branch twice. So I've had the honor of using your tank compressors for scuba dives. No, great questions. We haven't started that process, but that's why we're calling this universal biomineralization. We think that these fundamental universal principles will allow us to look at osteoporosis and be able to move forward on all these other types of manifestations of either growth or dissolution of the mineral deposits throughout we're making the same kind of headway on all the invertebrates on the coral reef as well. So we're doing the same work there. So thank you.